Okay, John. First of all, thank you very much for the invitation. We are not working on JCVI, but we certainly are inspired by the work that the various groups are doing in this direction. And perhaps our work is best described by what I've written here. That is the bottom-up design of synthetic cells based on simple components for requested function and their integration. And so some of the questions that we have in the group are, what are the requirements for a functional metabolic system in confinement? How can we build a fuel supply system? How can we obtain homeostasis? And how can we couple various reaction networks? And so a hallmark of life is that cells are selectively open and they operate out of equilibrium. And this is schematically shown here by this reaction network in a minimal cell, where there is a continuous feed of substrates and drain of products that allow the reactions to proceed. And it's these type of systems that we are trying to design. Well, for the feeding and draining of molecules, we take advantage of antiporter proteins. And an example is shown here that takes up a substrate, but in the back reaction, it transports the product out. We do this in synthetic vesicle systems, and an example is shown here. So the red molecule is taken up, delivers ATP, and the product, the blue molecule, goes out again. And so today we will be talking about how we can design and develop, construct networks to make ATP and electrochemical energy. I will talk about ATP and Mijer Patino after me about electrochemical energy. And we use these forms of metabolic energy to supply the cell with building blocks, to achieve homeostasis, to regulate the volume, expand the membrane, or in general, enable endergonic processes to proceed. Well, you're all familiar with how we in our body make ATP. So this is how it is done in mitochondria. So we respire, we form an electrochemical proton gradient, and we make ATP. Works very well, but it's very complex. It involves more than 100 proteins to do this and to assemble the cofactors. And so a much simpler way to make ATP is via the deamination of arginine, which yields the amino acid ornithine as an end product. And so these two molecules are antiported via this transport protein. Then there are three enzymatic steps that in the end yield ATP. And so in a vesicle network that we have developed, it looks like this: transporters in the membrane, enzymes encapsulated, product ornithine antiported out, and then we have small molecules like carbon dioxide and ammonia that diffuse passively across the membrane. We can determine the operation of the pathway, for instance, by determining the amount of ATP or the ATP-ADP ratio using this fluorescence-based sensor. And from one molecule of arginine, we can form one molecule of ATP. Well, this is an experimental result where you can see the ATP-ADP ratio here reported as a fluorescence ratio. And what you can see is when we add arginine, we rapidly form ATP, but then surprisingly, there is a drop in the ATP levels over time, even though we do not have ATP consuming reactions around. Also, we improved the system by having a fast antiporter, and the fast antiporter gives rise to a faster production of ATP, but also the drop in ATP is faster. And so this has been puzzling us for a long time, and so we've done lots of measurements, and we've built a mathematical model to describe the metabolism, and basically the solid lines that you see are actually the outcome of the modeling based on the experimental data. And what has gone into it, because we think there is a reasonable fit between the experimental data and the mathematical model, what goes into it, and that is kinetic parameters of the enzymes and transporters, concentrations of intermediates, equilibrium constants, stability of intermediates, futile hydrolysis, and the spontaneous formation of carbamate from CO2 and ammonia. And I'll show a few results, a few surprises that we have seen when we were characterizing this relatively simple pathway to make ATP. And then the first one is, and again, if you look at the reaction stoichiometry, we see from arginine we form one molecule of ATP, but we are consuming 3 protons. So you expect the internal pH to increase, and this is also what you see in the model. But if we do it experimentally, we look at it experimentally using this fluorescent pH indicator, then we see that there is an initial increase in pH, but then there is a drop. And that has been puzzling us. But if we then realize that, and I'm showing again here the full pathway, we then also realize that the first enzyme in the pathway, which deaminates arginine to citrulline, and then we found that citrulline can actually also be transported back out via this antiporter. What you then have is a futile hydrolysis of arginine, so the citrulline is exported out. For every arginine that is deaminated, we form ammonium ions, which are impermeable, but ammonia is permeable, leaving a proton behind, explaining the acidification that we are seeing at later time points. It also requires that, in fact, we are forming more citrulline than ornithine in this way, and that is also what you can see here. We form basically 4 times more citrulline than ornithine in this pathway if we run it under these conditions without ATP consumption. The model then also predicts that if we would consume the ATP, that most of this would not be happening. And how do we consume the ATP? In this case, we've used the mitochondrial ATP-ADP carrier, which transports ATP out and ADP back in. So this is the second antiporter system. Well, what you can see in the data here without ADP, so when this system is not functional, we again see the drop in ATP over time. However, if we add ADP, in this case after 90 minutes, we see a transient drop in the ATP level inside the vesicles, but then it recovers and remains stable for hours. In fact, it remains stable for at least a day. And so here we've reached a new steady state in which these systems are continuously producing ATP, but the ATP is also transported out in exchange for ADP. And so to summarize this part, we have a simple reaction network that basically functions as a mimic of mitochondria. But in order for it to function well, the produced ATP must also be consumed. And this minimizes the futile hydrolysis, it minimizes the internal acidification. It also minimizes the breakdown of carbamoyl phosphate, one of the intermediates. But also, surprisingly, because we form quite a bit of CO2 and ammonia, this then leads spontaneously in the formation of carbamate, and then carbamate turns out to be a substrate for the back reaction of the third enzyme. And then we have the futile hydrolysis of ATP to ADP, explaining the decrease in ATP levels at later times. So we have several, I would say, emergent properties in a pathway that is only composed of 4 to 5 components. Okay, can we do something useful with it? So can we couple the modules? And so what we did next, we used the same vesicle systems that excrete ATP, but now have the ATP taken up by secondary vesicles that consume ATP. And in this example that I will show, it has a transport protein that controls the volume of these vesicles. And so it consumes ATP, ADP is formed, and ADP then should go back to these vesicles and then being recycled. Here we show that we indeed excrete ATP by having the ATP-ADP sensor on the outside, and you can see we form ATP on the outside and it stays stable, the signal, over several hours. And then here we've actually used it, and so what we show here in this graph is massive amounts of transport of glycine betaine in vesicles B, so these vesicles, which depend on ATP production from arginine in vesicles A. And this is one of those experiments that when you design it, you hope that it works. And this is one of, I would say, one of the experiments you do in 5 years that works actually an order of magnitude better than you had hoped for. And so basically, the ATP generated in vesicles A fuels a reaction in vesicle B, which produces ADP that feeds back in vesicle A to keep this pathway running. What we also would like to do is to have the membranes grow. For that, we need lipid synthesis. And so one of the simplest lipids is diaryl phosphatidic acid, shown here, which can be synthesized from fatty acid, here shown as oleic acid, in the presence of ATP and glycerol 3-phosphate. And then we need to recycle coenzyme A. Again, the same vesicles, making ATP, excreting ATP via the mitochondrial ATP-ADP carrier, but now we also included an enzyme to synthesize glycerol 3-phosphate from glycerol. These membranes are highly permeable for glycerol, so we don't need a transporter for it, but they are highly impermeable for glycerol 3-phosphate. And so in order to get the glycerol 3-phosphate out, we need a third antiporter, which is the GLP protein. This one now excretes glycerol 3-phosphate, but takes phosphate back in, which is what we need to keep this pathway running. And so the glycerol 3-phosphate can now be used for the feeding, plus the ATP, for the feeding of the reaction network for the lipid synthesis. And what you can see here, we have lipid synthesis in these secondary vesicles, which is dependent of arginine in the ATP producing vesicles. And so we have a simple reaction network that functionally mimics mitochondria. I've already shown these emergent properties. We use several antiporters to maintain out of equilibrium conditions, and we can couple the ATP production to the consumption in vesicles, for instance, for the synthesis of lipids. And so we have an energy conservation module that really works well. We have it optimized, we have it under control, we have it captured in a mathematical model, and we have seen it with unforeseen and emergent properties. And of course, this is the most exciting part of the work because it brings us a better understanding. Well, these vesicle systems we have so far studied in solution. And what we are doing right now is to encapsulate them into larger vesicles, as if we would have a mitochondrion in a mammalian cell. These vesicles produce ATP, enable ATP-consuming reactions, but of course in such a system we also need homeostasis, for which we need an electrochemical module to generate the protomotor force. And in the next talk, Mijer will show how very simple decarboxylation reactions can perform this. And then after that talk, Jelmer Kunradij will present the properties of the membrane of these vesicles in comparison to the properties of the membranes of these larger vesicles, which is important to get these reaction networks coupled to actually feed the vesicles with arginine, produce ATP from it to enable other processes to occur. And so this is my last slide. I would like to thank several people. I've highlighted them in color, and what they have done matches with the different modules that we have made. And specifically, I would like to mention Jelmer Kunradij and Henry Hoppe. Jelmer will present their work, and Mijer Patino and Zaid Ansari, and Mijer will present their work. And with that, I would like to stop and thank you.
Questions for Bert? I'll start. It seems to me that, again, I come at this from a minimal cell centric view, but imagine if you wanted to take our really simple system and using some of the things that you've talked about here, engineer it to be a mammalian cell symbiont so that it could function a little like a mitochondria. Granted, maybe not as efficient as a mitochondria, but as a tool to export, to push ATP out into the media of a cell. Does that seem plausible?
Yeah, definitely. I would say this system can be as efficient as mitochondria, except that we are less flexible. We can only use arginine. In principle, a mitochondrion can oxidize many different compounds through all this complicated process of respiration. We can then still make ATP. But no, definitely, we could use it for various functions. One of the things that we are also doing is actually to get a better understanding of the process of cell division and use these, let's say, mitochondrial mimics to drive processes, and importantly, maintain out of equilibrium conditions. So constant ATP ratio over periods of hours to days, because that is often what we need if we want to study or develop a process.
John, you're on mute.
Oh.
Hey, John, Kim here. I got another question. Bert, this is really nice to meet you, and this is really interesting work. Just for your information, you know, the cell from which the minimal cell is derived had the whole arginine deaminase system in it, actually -1 ARC-A, which we put back in, and it seems to function. But we just simply got rid of it because it was not essential to make the minimal cell, the Syn3A. But if you ever needed to put this system back into Syn3A, it should be quite straightforward. We thought perhaps in the beginning we could leave it in. The only ATP generating system in the minimal cell, the Syn3, is glycolysis, but we thought perhaps putting ADI back in would give us some extra ATP. But I'm not sure from your modeling that's true. It would be interesting because that ATP, if it has to be used for other things, would it be sort of a futile generation? So I'm just sort of throwing this out because it may be interesting if you ever wanted to have a construct of the minimal cell with the system in it. It should be quite easily accomplished.
Yeah. No, so these enzymes are present in many cells. In fact, the second enzyme is also present in the cells of our body, and where it is involved in the opposite reaction, producing arginine.
Right.
So what we have done also is, I know your minimal cell uses the glycolytic pathway, and that also provides the cell with a lot of ATP. For us, that is a more complex pathway than this one. And so what we also have done in the meantime, in the collaboration with the group at Wageningen University, we've made an E. coli cell that has become dependent for its ATP production on this pathway. And so what we are doing right now is we are actually evolving that strain to actually improve the pathway further in E. coli by selecting for faster growth of the E. coli on ATP produced by by this pathway. Yeah, so in principle, if we can do it in E. coli, then it should also be possible to make JCVI even more minimal, simple in terms of components, and have ATP made from arginine. Of course, the glycolytic pathway is also used for other intermediates. So it's also used for sugar intermediates, for, let's say, for the pentose pathway and for as building blocks for amino acids. And of course, we would have to supply them these then separately. But in principle, this pathway could replace something like glycolysis.
That'd be interesting.
It would be really straightforward to use our landing pad to drop just a handful of genes into JCVI Syn3B that would give you a system that did the reactions that you're showing there in a way that would be much simpler than what you're working with E. coli.
Yeah.
Oh, sorry, Bert, I didn't mean to interrupt you, but like we did it, John. In the lab, we inserted the adenine pathway and actually it works. It would be very interesting to test. Like I was thinking about your vesicles with more ATP together with them and see it would. I will be presenting this work actually on next week, so it'd be interesting to see how we like. I think something of your results maybe will bring some other insights in our results, in what we are aiming to do. So this is very interesting. But yes, this the pathway works on Syn3B.
Yeah. Well, what I would also like to emphasize, we started this to make ATP. What is very interesting is that the pathway, depending on the condition, can behave differently, and it can acidify. It can have these futile hydrolysis. We hadn't realized that when you produce a lot of CO2 and ammonia, that you form carbamate. And carbamate is, in fact, as I mentioned, a substrate for the third enzyme driving the back reaction. Carbamate is also a precursor for carbamylation. And carbamylation is a chemical modification that is affecting proteins, which is something that we are likely seeing in our system as well. So basically, we have obtained quite a bit of understanding and also things that have puzzled us for a long time when we were developing the system. And that would be more difficult to do in E. coli or a minimal cell, because that is still, I would say, a few orders of magnitude more complex than the system that we have. And I think we are almost ready to move back to perhaps JCVI and test some of the things that have come from these in vitro experiments and test them in your wonderful in vivo system.
Super. Okay.
Can I do one last experimental question for Bert? How do you get the transmembrane channel ARC-B into the synthetic membranes? Does that work?
Okay.
I mean, yeah, and is it the right orientation and function?
Yeah, no, very good point. What we do is we do detergent-mediated reconstitution. And so I'm not going to mention the details. The orientation of most of the proteins is random, so 50% in and 50% out, compared to what they would be in vivo. But the beauty of these antiport systems is that they operate equally well, irrespective of whether they are oriented inside out or outside in. And that has to do with the fact that the transport is determined by the electrochemical gradients and not by the orientation of the proteins.
Yeah, very nice. Yeah.
And so the electrochemical gradient drives the antiporter, as opposed to having it being, I mean, it's a passive system as opposed to requiring energy. Yes?
Yeah, yes. But let's say if you have arginine on the outside, you have a lot of arginine, you have a low concentration on the inside because it's metabolized, then on the inside you form ornithine, and you have a gradient from ornithine from in to out. And so basically the inward gradient of arginine and the outward gradient of ornithine drive it. But you don't need ATP, you don't need a protomotor force for it. It's the own gradients and the metabolism that actually drive the processes.
I understand.